Hello, and thank you for joining us today. You'll hear from two chief scientists on combining human-on-a-chip technology with PBPK modeling for in vitro and vivo PK/PD extrapolation. Joining the panel is Dr. Jay Hickman from Hesperos, the human-on-a-chip company, and Dr. Viera Lukachova, chief scientist from Simulations Plus. Now, before I turn things over to today's presenters, a few housekeeping notes. We take your privacy rights seriously, and by attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. Immediately following today's presentation, there'll be an opportunity to ask questions. We welcome and encourage you to submit your questions at any time during the presentations using the question panel on your dashboard. And if you need assistance during the webinar, please use the hand raise icon and our staff will assist you. Now, a quick getting to know you poll while everyone connects. Which sector best describes you? We'll give you just a few seconds to go ahead and collect your answers. And once again, thank you for joining us. And I'd like to go ahead and turn things over to Dr. Hickman. You should be presenter now.
So can you see
And
my
I
screen?
confirm we can see your screen. Thanks, Jay.
Can you hear me okay?
Yes.
All right. So trying to get the screen to minimize. Oh, there we go. So Hesperos is a small biotech company located in Orlando. Founded with Mike Schuler and I about 5-6 years ago. We licensed a bunch of patents from UCF and Cornell. We won the Lush Prize for animal testing in 2015. We moved into a new 14,000 square foot facility 2019. And the company right now is about 32 at present, and we're actively recruiting. We're a service-based only company. We don't have any plans in the near future to offer the systems for sale. The main thing I really want to talk about today, and it shows you the relevance for in vitro, in vivo translation, is we really have focused on this idea of clinically relevant functional readouts. And that what we're really talking about is when you walk into a doctor's office, he doesn't immediately stick a needle in your arm to be able to check for biomarkers. He looks, how are you walking? How are you talking? Are you making any sense? He checks your reflexes by smacking your knee. These are all electrical, clinically relevant functional readouts. We can also look at barrier integrity for things like the blood-brain barrier, kidney systems, and GI tract. So we can also monitor all these systems non-invasively. So what we can do, we don't have to just take it apart to be able to digest the cells, to be able to understand what the biomarkers are. And this allows us to also chronically monitor what's going on. It also reduces substantially, if not eliminate, the need for biomarkers in a lot of experiments. If you had to buy a per mega kit, you know how expensive that can be. But also can allow mechanistic determination for toxicity and also for target identification. So this is sort of a very busy slide here where this sort of describes the original process Mike Schuler did when he came up with the first patents for this back in the 90s, was he took a PBPK model, and which most of you will know if you're on this call, in that looking at the equations that are used to calculate the dosing for phase 1 clinical trial, and said I can modify those and then design a chip. This is one of our more advanced systems right now where we're looking at overdose. I'll talk a little about that later. Models for opioids and also recovery with naloxone. And these are all the references to be able to build this. But the key point for this multi-organ system is it's pumpless. What that means is we can actually use gravity to drive it. We just put it on a rocker platform and use that. What that does is it eliminates all the tubing, all the pumps, and also makes it a low volume system. So I can, if I have the appropriately sized liver, I can be able to look at the metabolite and parent in the same system at the same time. And so this is a very busy slide here, but I've only got about 25 minutes, so I can't get into all the details. But it just shows you how we build one of these systems, in this case for cardiac. So what we can use the surface chemistry that we do to be able to try and pattern, in this case, cardiomyocytes, but we can do it with neurons too, over top of microelectrodes. So we can actually then stimulate one and then monitor what's going on in the other electrodes. We can look at rhythm generation, conduction velocity, and AP length or QT interval in the same system. Now, in a separate system, we can actually measure force, in this case, with cardiomyocytes, but also for skeletal muscle, where we have a very thin piece of silicon here. So when this actually is contracting, we can measure the bending with a photodetector, and then we can actually then look at that deflection and calculate force. And that gives us our cardiac output. And this is actually what it looks like with unpatterned cardiomyocytes. So if you have a event here which is causing stimulation, you can then, but what happens is it's very disorganized tissue that you get dispersion losses if it moves further away. Whereas you can see here on the cantilevers and also on the patterns, you get lining up of the sarcomeres and the muscle. So whether or not it's a short path length or long path length, we pattern it, we get the same conduction velocity, which allows us to measure spontaneously the propagation of action potentials, but also more and better, we can stimulate and stimulate and actually say stimulate here and watch it going over each electrode. And at each point here, we can actually measure the QT interval of the system. So we've done well over 50 cardiac influencing drugs with this system. I'm going to focus on 3 to just prove a point, which we can actually then hit it with norepinephrine and speed it up. We can do this in a dose response curve manner. We can look at verapamil, which actually is a calcium channel blocker, and we see the reduction in force. And then sotalol causes a QT interval change, which we can measure and be able to get a dose response curve for that, but it doesn't affect the force. So now if we didn't know what the toxicity mechanism was or the efficacy mechanism was, we could actually ignore all the different pathways having to do with force and focus on electrical. And there's other things we can do to then try to narrow that down even further to get it to specific pathways or potentially targets. We can then take that cardiac system and then add a liver system to it. This is actually liver cells in our system going out in a 2D for 7 days and also out to 28 days. Very good morphology in both 2D and 3D systems. And we were able to show that the CYP activity was maintained, as well as albumin and urea production at a standard level as seen with the best in vitro systems, and showing that we can maintain this for 4 weeks or 28 days. And this was published in Biotechnology Progress recently. So we can then, like I said, combine these, the chips for the cardiac force, the cardiac electrical activity with a liver-based chip. And these are the recording systems that we have for it. Now, and again, we could do simulation design. So again, if you came in and said, you know, what we're focusing on is custom building systems. So here we can actually design it to be able to look at the, simulate the flow rates, and then also be able to get the shear stress in no matter what configuration we have. And this is the resultant system here. I can show you a picture. And then a project we did with AstraZeneca, we looked at as a control, we wanted to look at the terifenadine-fexofenadine couple. Terifenadine, which is the parent, is toxic and was pulled off the market for QT elongation. You can see in a cardiac-only system, we see that toxicity. When we add a liver into it, it's a metabolite, fexofenadine, is not cardiotoxic. It is actually on the market right now, I think is Allegra. You can see it's a very large shift for QT interval. Conduction velocity is not affected with the liver there, but with cardiac only, you see a IC50 about 32 micromolar. And again, the most important point is we're trying to do clinically relevant functional readouts. So viability should not be affected if we're trying to relate this what's going on clinically, and the viability is not affected, even though we're seeing the functional measures being affected quite strongly. So again, we can actually model the system, and we can then take the account of the absorption of the compound onto the housing materials. We can look at low absorbing materials like fexofenadine, high absorbing materials like terifenadine. We can actually then simulate the mixing. We can actually then simulate the loss, which is the conversion of parent to metabolite, but also the loss of any absorption into the housing materials to be able to get the area under the curve. So everything we get is the actual exposure and not the as-dosed concentrations. So we can actually then take this out for, in this case, 14 days and do the simulation for fexofenadine, the metabolite, and for the parent, and be able to show by HPLC-MS that we get a very good agreement between the modeling and the actual in vitro results. We can actually then take this, we did this in collaboration again with AstraZeneca, and be able, and then Vera will expand upon this in her portion of the talk. So we can actually then account for the PDMS absorption loss in the system. We can account for the conversion. We can actually then look at the exposure of the outside of the cardiac cells, and then be able to get the PK of the system and be able to predict it very accurately, as you can see. And then be able to see what is transported across the membrane, to be able to affect the FPD in this case, and be able to look at the PD, and again, be able to accurately be able to predict this as well. And to the point where we were able to predict very well the in vivo data they had for guinea pig, dog, and non-human primate. And again, this was published in Nature Scientific Reports in 2019. And again, this is the real value of what we can do, is we can extrapolate the in vitro results to be able to predict in vivo PK and PD in the systems. Now I'll be able to show you some more advanced systems in terms of what we can do in terms of the in vitro modeling of the systems. Like I said, we can very easily custom design systems. So Roche came in and said, well, that's a really nice cardiac liver system. Can you actually add in cancer cells? And we're going to make it difficult. We're going to actually look at the difference between a multi-drug resistant cancer and a non-multi-drug resistant cancer. They wanted to look at tamoxifen. Tamoxifen by itself is not a great chemotherapeutic, but its metabolite is. So we saw tamoxifen without liver really didn't affect the cardiac proliferation, but with the presence of the liver, it did. When we treated it directly with only the metabolite, we saw about the same. For the multi-drug resistant cancer, with liver, without liver, with liver, and the metabolite really didn't do anything as it was expected. But the reason these cells are multi-drug resistant, it has a pump called the PGP pump, which pumps out the chemotherapeutic as fast as it goes in. But if you block that, in this case, which we did with verapamil, you can actually then see a reduction in proliferation. But when we add the verapamil to the non-multi-drug resistant cancer, we can see that it was actually blocking the channel. We don't see any increased proliferation loss with that system. Now, if you monitor the off-target toxicity, again, this is exactly the same system. We're adding the tamoxifen to the recirculating medium that is going around all the different organ systems at the same time. So we can actually see the presence of the parent and the metabolite. We see that tamoxifen causes a decrease in conduction velocity, but recovery. But when you add verapamil, you see from the drug-drug interaction, you see a reduction, and after 3 days, no recovery. Cardiac force, same thing. With tamoxifen by itself, you see recovery, but very little recovery with the drug-drug interaction. But for beat frequency, you see it go down and actually recover, and you see that also as well. But again, cardiac viability was not affected in the system. Liver function was also not affected. We're looking at albumin, urea, and viability was not affected the liver, but we knew it was fully functioning because we were seeing the effects of the metabolite in the system. And this was published in Science Translational Medicine 2 years ago with Roche. We can also build neuronal systems, in this case, a neuromuscular junction. We can actually put on one side of a barrier, we can actually create put the motor neuron cell bodies. We can then put the muscle and grow them into myotubes on the other side and grow the axons through the tunnels to be able to integrate on the other side. You can clearly see the axons going through the tunnel and forming neuromuscular junctions. We published the first dose response curve for Botox with an in vitro system, human in vitro system, which agreed very well with the doses that they had on the vial. This was calculated using our system and published in Biomaterials in 2018. And again, this was the first example of a human-based toxicity description for Botox, which is normally all done in animals. But we're really interested in using these also as disease models. So we have an ALS project, Lou Gehrig's disease. So we can take stem cells from the patient, differentiate them out with the mutations, and then be able to build the same system. And actually, in this particular case, instead of using the cantilevers to measure force, we're doing motion capture, where you can see the wild type as we stimulate the motor neurons. We then can actually see the contraction of the myotubes. You can then correlate that with the simulation and the force generation. You can then see for the mutation, in this case a FUS mutation, you see a loss of fidelity in the system, which you get skipping. And we can actually saw this is reversed with a therapeutic treatment. And again, this is completely clinically relevant because we talked to somebody who was doing a clinical trial, and they said, well, what are some of the tests you actually do? So one of the ones we do is we have a patient just do a task faster and faster. What we see shaking, we see jittery, and we see loss of strength. So we can actually then test for that by just stimulating the muscle, the motor neurons faster and faster to drive it into tetanus. And what we can see is that for the wild type, again, you see a marry for each time we stimulate, we see a contraction. For the SOD1 mutation, you see a variance in the system. You can also see that, depending upon the mutation, you get different effects. In this case, you get loss of tetanus. And then FUS, you again see that the effect here, and again, not really great. You get unfused tetanus in the system as well. We actually have the control. We can actually go in and then stimulate the motor neuron, the muscle directly. And as you see, the myotubes are all functioning perfectly, showing that the defect is at the neuromuscular junction. And actually then looking at the different frequencies, you can see this falls off for the mutations at day 14 and day 17. But also another clinical correlate is fatigue index. And this, again, something they do for the clinical trial work. And again, really what you're doing is looking at the tetanus and ability to maintain that. And we see for the disease model that the mutations were not able to maintain this nearly as well, okay, as the wild type. And what we did is we then took a therapeutic treatment with the Vienna protocol, which is a protocol for ALS patients developed by a doctor for his daughter. Anecdotally, had been seen to be very effective for over 1,000 patients through their website. We were able to show that that actually was able to give some rescue of the effect at day 14, but also at day 17, and not so much at day 14 for the fatigue index, but by day 17, you get a very robust rescue of the system. So we can actually then create a model-based system by taking diseased cells from a patient and differentiate out the stem cells into the organ that is most affected, and then be able to create disease models. And especially this is for rare diseases, where in many cases you have about 7,700 rare diseases, but there's only about 400 active research programs because there just isn't an efficacy model for it. Whereas we can actually, we're working with a number of different companies right now to be able to custom build these systems for disease models for rare diseases. And we can also then take sera samples for things that actually cause complement deposition, and then show that we can actually reverse those effects as well and create a patient-specific model by just taking their sera versus actually having to grow out the stem cells as well. We're also interested in looking at CNS models. We want to be able to look at a model of long-term potentiation as a fundamental unit in learning and memory, and especially for something like Alzheimer's, it is what the main deficit is in the fundamental ability to learning and memory. So we can also pattern the neurons in the system. So this is a neuronal cell here. This is the only rat data I'm going to show you, but it's the best slide we ever got out of this. And it's really cool to be able to see the putting out the processes that actually then explore the somal adhesion site, the translocation after communication back and forth, and then puts out the neurites to become the axons and dendrites. And we can use this surface patterning technology to actually build circuits, which is then very important for looking at diseases like Alzheimer's, where we want to be able to stimulate one electrode and see what happens on the other electrode. And then we can do that very robustly. We've actually been able to create a model for Alzheimer's by treating it with the oligomers for A beta. And this is the case where we actually were able to stimulate and were able to induce long-term potentiation, which was maintained after treating with A beta scrambled, but abolished after treating with the oligomers. And also seeing the same thing for tau oligomers, where that was maintained with the control, but abolished with the tau oligomers. And that was published in Alzheimer's and Dementia, Translational Research and Clinical Innovations this past year. Again, we can combine these all together to be able to put cardiac mechanical, cardiac electrical, skeletal muscle, neuronal, and liver all in the same system. We can then model that. This was published in 2016 with the initial paper, but in 2019, we were able to take these systems out for 28 days. These are the figures from that paper showing very good liver morphology out to 28 days, very good albumin production, urea production, and CYP activity maintained in a multi-organ system with recirculating media. Again, cardiac contractile force, you can see the cardiomyocytes on the cantilevers still there after 28 days. Very consistent force behavior. Looking at conductive velocity in the system, again, you can see the cardiomyocytes over the electrodes. You can see all the different parameters, beat frequency, QT interval, conductive velocity, and MISI was another way of measuring QT interval in the system. You can see the muscle contractile force. You can see the myotubes on the cantilevers, and they've been very consistent behavior. And then this is motor neurons patterned over top of the microelectrodes. And you can see here that the nice spike amplitude and the spontaneous activity goes up with the 4-organ system for 28 days. Now, what we can also do, not only looking at acute drug dosing, we can also look at chronic drug dosing. Most drugs are meant to be taken over a period of time. And so again, we can model this and be able to look at see what the dosing in our system should be and be able to look at chronic versus acute. It's just one experiment we did with doxorubicin in collaboration with L'Oreal to be able to look at chronic drug dosing. You can see here that the conduction velocity goes down, okay, almost immediately with doxorubicin. The force is maintained for a little bit, then starts to go down. And the number of cantilevers also starts going down as well. But if you then look at the morphology of the system, you really see no effect on the morphology out to day 9, okay? And so we're actually right here, we're actually seeing a very large change in force. And then doxorubicin starts to cause some morphological changes after day 11, where then the pattern is actually lost at day 14. But if actually you're monitoring beat frequency, these things are all still beating. So this is a much more sensitive measure, conduction velocity and force, than it would be for looking at beat frequency. Lastly, we are going to go through the modeling we can do for an overdose model. I mentioned that earlier, where we've been able to differentiate the pre-BOT-C neurons, human neurons, with the pre-BOT-C complex in your brainstem is what controls rhythmic breathing, which is what opioids basically interact with to cause a cessation in breathing, which is the main reason that people have overdoses. But we wanted to see not only create a dose response curve for the pre-BOT-C neurons, and so it's rescued by naloxone, as they do in vivo and in clinically, but also look at the off-target toxicity of repeat dosing of naloxone. I want to show we actually had the pre-BOT-C neurons first, so these are all the markers to show that we can actually then differentiate them into the pre-BOT-C neurons, and these are the markers NK1R and the mu opioid receptor that show that it's co-localizer, so these are pre-BOT-C neurons, and also that they're electrically are activated by ATP and substance P, and SST turns off the activity, which is exactly what you'll be able to see for pre-BOT-C neurons. And be able to show for an opioid, Damgo in this case, with a synthetic opioid, it actually decreases the firing frequency. We can get a dose response curve for that system and be able to, we can actually do dose response, and it was rescued by the treatment with naloxone in the system. We also published a paper with Roche again, looking at the immune system. We can put monocytes in the system and be able to monitor their cytokine profiles when we actually then selectively damage a organ or activate the entire immune complex, like create a cytokine storm by treating with bacterial proteins. And we're able to see the signatures for a M1 restorative macrophage versus an M2, which is a inflammatory macrophage. And this was published in Advanced Science in 2020 in the system. And then lastly, we can also integrate barrier tissues with these systems. In this case, we mentioned working with L'Oreal, which is a skin mimic system. We can also introduce GI tract as a toxicity target, not functional yet. Blood-brain barrier as a functional system, and then GI tract as a functional system as well. So I already mentioned the whole idea in terms of, what can you do with these systems in terms of rare diseases, but also people have discussed reducing the number of people, say, in a phase 3 clinical trial, and also to be able to take and steer preclinically the use of companion diagnostics to be able to limit a clinical population. Again, reduce the number of phase 3 by creating human variants. So if you know what are sensitive variants that might cause problems in terms of the general population, the outliers, you could actually look at these uncommon genome profiles in the in vitro systems where they're versus having to try to find them with a phase 3 clinical trial. Because remember, Vioxx was pulled off the market because it still showed cardiac side effects that led to death, even after doing a 30,000 person clinical trial. Already talked about the idea of adding in vitro clinical trials for rare diseases to be able to lead into INDs, which we've already done with MMN and CIDP with BioVerita, which was filed this past year. And also, they've also talked about the idea of, if you already have safety data, potentially going directly into patients with these systems. Better evaluations for children, aged individuals, and obviously the cancer work that I showed you would be something we're looking at for individualized for cancer patients to be able to figure out what is the best chemotherapeutic regimen, both for not only parent but metabolites for both efficacy and off-target toxicity. So lastly, this is the team, not only at Hesperos, but also the collaborators at UCF. And at this point, I turn it over to Vienna.
Thank you, Jay, for this great overview of the technology and the flexibility that it offers in terms of combining the different tissue types depending on the exploration, as well as variety of different PD aspects and effects that can be simulated or explored with this technology. So let me make sure I'm showing the correct screen. Right.
Oh, yep, we see you in
Hopefully
full
everybody
presentation.
can see the slides.
We can.
Yes. So in this second portion of this talk, we are going to focus a little bit more at possibility of modeling these with the PBPK model. This is actually screenshot from one of the publications that Jay also referred to, that terfenadine, fexofenadine modeling, looking at the cardiac effects, right? And how modeling the system, the human on a chip system with the liver and heart tissues, right? Processed it using a compartmental type model, right? And then fitted the PD model, linking the concentrations in the cardiac tissue with the effect, right? And then using this PD model to predict what might be the PD effect or the toxicity in the in vivo. Within this application, this extrapolation relied on having observed PK profiles, so experimental data either in preclinical species or in human. And again, that effect was then linked to the plasma concentrations. So looking at this, we felt that there is a lot of information that actually might be wasted through this part, right? If we are not doing a full PBPK model, trying to explore also the distribution mechanisms, right? And focusing only on PD effect. So using a PBPK model or mechanistic simulation of these models, this in vitro system, and then linking it to PBPK model could give us a best of both worlds, right? Where the PBPK models are already used for heavily and reliably for first-in-human PK predictions. This technology offers a promise for doing a first-in-human PD predictions. So can we just do the full mechanistic model and PBPK to do both PK and PD before doing any studies? So we've started looking into this. I'll start with just a very brief overview of the PBPK model, what we are talking about here, right? Of course, we are talking about describing the physiology with individual compartments, where these compartments represent individual tissues in the body, and they are linked to a blood flow. The blue arrows are showing the blood flow in the body. Right? And each of these compartments represents a specific tissue in the body with its own physiological volume, blood flow, expression levels of enzymes and transporters, composition of that tissue that drives the distribution of the compound between the plasma and the tissue itself. And these distributions would be dependent, of course, not only on the tissue composition, but also on the compound properties in terms of its lipophilicity, ionization state, binding to the proteins, and binding to the acidic phospholipids or blood to plasma concentration ratio. And as the blood with the drug within it moves through the tissue, of course, the distribution through that specific tissue could be described as a simple partitioning process. If the drug diffusion through the cell membranes is very quick, and it's only about partitioning between the plasma and the tissue space itself. This is then driven by the tissue plasma partition coefficients. And we already have a number of methods to predict those KPs or tissue plasma partition coefficients from the tissue and compound properties. And within any of these tissues, there could be clearance or elimination of the compound as well. Of course, if the compound has more difficulties moving across the cell membranes, or potentially is a substrate for any transporters, right? We may need to employ this type of more complex model to describe the distribution in the tissue, where the compound would be quickly partitioning only between the plasma and the extracellular tissue space, but then would be either slowly diffusing between the extracellular and intracellular space, or would be subject to the movement by transporters, either uptake or efflux or both. So looking at this, similar processes could be applicable for the distribution within that human on a chip model. And this actually brings us back to the origin of this human on a chip model, as Jay was explaining, right, where this came from, that they looked at the PBPK model, right, and decided to put it on a plate. So what we are trying to see here, can we now complete the circle by now modeling this and putting the data back into the PBPK model? Again, this is just the schematics and what would be the schematic representation of the flows and the tissues going or within this type of mechanistic model, where within this setup, we have two reservoir compartments. Right? We've got those two tissues, and this one was specifically for that liver cardiac setting, where this would represent the liver tissue. These would be the two cardiac tissues, right? And the In this case, we have double-headed arrows specific for this type of setup where the flow of the media is driven by the basically gradient and flow as the plate is getting tilted. So in this case, instead of flow going in the circle, like in vivo model, in this case, it is more as a rocking motion. So the media is flowing back and forth. But again, each of these compartments would be represented by its own specific volume. Right? Of course, much smaller than in vivo, but still a specific volume that could be defined. The flow pattern is slightly different, right? Or in some cases, significantly different than in vivo, but still could be described mathematically and look at what we have, the flow of media through individual compartments to look at the distribution. Since these are specific tissues or the compartments representing or mimicking individual tissues in the body, the initial assumption would be that the enzyme and transporter expression levels are going to be close to the expression levels in vivo, even though this needs further work and verification with the standard substrates to be able to verify that the activities are really the same. Since they are grown as an actual tissue cells and functional tissue cells, again, we would be assuming that the composition of those tissues in terms of how much lipid, how much water, how much protein they have, would be very similar to the one in vivo, which would simplify the scaling of the partition coefficients. So under these assumptions, again, distribution could be handled using similar models, right? We could even potentially derive a method for the KP media, right, by accounting for the differences in the media flowing within these cells versus the plasma that is in the real in vivo situation. So if we compare these two models, right, the full PBPK model for in vivo simulation and this type of mechanistic model for these in vitro models for these in vitro experiments, right, the aspects that we need to account for are, of course, the potential differences in the flow of the media, as I was already describing, where in vivo, we are basically looking at the media flow in circle. While in this case, especially at least with this specific setup, with this pumpless system, it is more of a rocking going back and forth. The distribution into the tissues and metabolism mechanisms are expected to be similar, so that should simplify the in vitro to in vivo extrapolation. The additional aspect that Jay already mentioned is that needs to be considered for these in vitro systems is the additional binding process. Potential binding to the materials in those plates. But again, this is something that can be easily incorporated in a mechanistic model to account for as a separate process that would be affecting the concentration gradients, would be affecting the distribution into the tissues as well as PD effect. So for translating individual parameters, right, when it comes to the tissue plasma partition coefficients, with the very initial assumption that it's the serum-free media that's circulating in the system, we could start with an initial assumption that perhaps the KPU would stay the same, or that the system would be giving us an estimate of the KPU value, since there is no protein binding in the media, right? And that could be then easily translated into KP value in vivo. Right? Of course, depending on the exact composition of that media, this could be modified to account for the processes of partitioning. If we have a more of a permeability limited type distribution, right, where we need to account for a slow diffusion through the cell membranes in specific tissue, right, again, we can fit the specific or the permeability surface area product for the in vitro system and use the strategy that we have now verified for a variety of different in vivo simulations, the strategy or the concept of specific PSTC value, right, or this permeability surface area per milliliter of tissue volume, right, and scale that from the in vitro to in vivo. Again, under this assumption, since we know what is the volume of that media in the in vitro system, what is the full, volume of the tissue in the in vitro system versus the full volume of the tissue in vivo, right? This could be a simple scaling from the in vitro to in vivo. And again, this I already mentioned, that the initial assumption is that the metabolism or the carrier-mediated influx, efflux, at least on the basolateral side of the cells, would remain the same. But of course, this is something that we need to verify with the experiments against standard compounds. Right? Potentially, this system would allow us to also link this with other in vitro experiments. Right? Let's say we have a complex system where a compound is a substrate for transporters, right? But is also metabolized in the tissue in the system, right? If we can, let's say, we have a liver tissue and we are dealing with the uptake as well as metabolism, right? Again, we could use metabolism or parametrize metabolism from human liver microsomes, right, as a simplified system for metabolism alone. And then this system combining everything would then allow us to more accurately separate out these two aspects and more accurately parameterize the transporter effects. So it does allow that type of flexibility. So enough with just the theoretical talking about how we would go around about this modeling, and to show some of the results from the preliminary models. I do need to emphasize that this is a preliminary model because we have not actually built or coded yet the full system accounting for the actual flow of the media in these systems. What we've done for now is just the initial sort of a exploratory simulations where we adjusted our PBPK model, right, by simply removing the extra tissues and including only the heart and liver tissue as we need to for the from the PBPK model, right? As a very simplified description, just to see is there something that could be done with these extrapolations. And this is showing the initial modeling again with this preliminary model with the approximate flow of the media for that terfenadine, fexofenadine model. And here I'm showing only the terfenadine results. On the left, I'm showing the results from the system containing only heart tissue. And the blue curve is showing the media concentrations. The brown is showing the heart concentrations. So in this case, looking at the shapes of the profiles, it is very clear that we might have to deal with some permeability limitation or with the more complex process of drug getting into the heart tissue. So we modeled it with a permeability limited tissue and fitted PSTC. And we incorporated a non-specific binding or slower non-specific binding to that would represent a binding to the plates, right, to account for this drop in the concentrations over time. Since this was only heart tissue, right, the metabolism is not expected to be as significant as when we add the liver tissue. So a lot of these drop in both concentrations is actually likely due to binding of the compound to the plates. So using the same parameters for these processes, right, then adding the liver tissue into the model, right, we now fitted the KP for the distribution into the liver tissue, right, and the metabolism by 3A4. Right? Terfenadine is assumed to be metabolized quickly and nearly completely by 3A4. Right? So since we have only one concentration level at this point, right, we approximated or we used the Km value as predicted by ADMET predictor and simply fitted the Vmax value to match the exposure. Right? And as you would expect, with addition of the liver, which seems to be accumulating the compound a lot, but also contributes to significant clearance, metabolism, right? The concentration in the media dropped much faster, and of course, much less compound would get into heart tissue. And once we had these simulated heart concentrations from both sets of systems, of course, then the last step would be linking this heart concentrations into the PD effect with the PD model. But this is the portion that can be done, and that is already done typically also if the full PK of the distribution is modeled by a simplified compartmental PK model. So the main focus was then, now that we have a model that, or parameters that reasonably describe the distribution within the system, can we actually predict what would be happening in vivo for terfenadine in terms of PK? Again, it's a preliminary model, so some parts worked, other parts not so great. To walk very quickly through how this extrapolation was done. Right? Again, as explaining earlier, the assumption was of a serum-free media, so no or minimal protein binding in the media, so assuming that the in vitro value gave us Kpu value for liver. Right? So that was simply recalculated to Kp value for in vivo. Of course, this only gave us liver Kp, and for the full in vivo distribution, we need the whole or the Kps for all the tissues. So at this point, having an estimate for liver Kp, we simply explored which predictive method would give us the closest prediction of that liver Kp, and use that method for predicting the distribution into all the tissues. Into all the tissues except for heart, where we, based on the in vitro experiment, there was the indication that it might be a slower diffusion, right? So here, where we did the distribution with the PSTC scaled from that in vitro system. A lot of other properties predicted either by the Membrane Plus software to account for binding in enterocytes, or by ADMET predictor. So the last part was the clearance prediction. And this is where we did have the difficulty. This is the part that did not work so great from a pure in vitro to in vivo, where in order to match that very quick metabolism in vivo, we did need to increase the Vmax significantly. And this did include, and this does also include the intestinal or the gut metabolism. Since we had the estimates for the Km Vmax values from ADMET predictor and the fitted value from in vitro system. And we do know, or we do have in GastroPlus built in the expression levels in liver as well as in the intestine. Right? It was easy to extrapolate from that liver 3A4 metabolism to estimate the intestinal metabolism. Right? But even incorporation of the intestinal metabolism still significantly underpredicted the clearance, and we had to increase this. Of course, there are a number of aspects that could be contributing to this. One that I was already mentioning a few times is that currently we were considering only approximate media flow. And you might have noticed in one of the slides that Jay was showing when they were modeling the flow of the media and the distribution of the compound, right? That might have significant effect and significant impact on how the compound was actually metabolized in vitro. Did the significant binding to the plates mask some of the metabolism, right? Again, something that could be explored as a separate process. Is there potentially different activity of these enzymes, even though this is less likely considering all the explorations and characterizations that Jay was already showing about functional tissues and how they really are matching what would be happening in vivo. So this is probably the least likely scenario, but again, something that could be explored with standard substrates for these different enzymes to evaluate whether the activity is the same or whether there is some scaling factor needs to be included. Or maybe there was a different rate limiting step that we still didn't incorporate for this compound. So it's not perfect yet, right? There is work to be done, but based on what we've done so far, it does show a promise, it does show a potential. Right? And the final step that I forgot to highlight, once we model the in vivo situation with a PBPK model, right? Then even for the in vivo prediction of PD effect, we can now link that PD effect to the actual concentration in that specific tissue. Right? Don't have to rely on the plasma concentrations, but can link it to that specific tissue, just like it was built from the in vitro system. So to sum this up, the model, even though we still have, as I mentioned, some uncertainties, we believe it does have potential for the full PK and PD extrapolation for first-in-human dosing. Right? The use of PBPK model allows linking the PD effect then to a specific tissue concentration. Right? The preliminary simulations show that there is a good potential for predicting the tissue distribution. The part that we need to work out is the clearance prediction, right? Why that one did not work, and really see whether it is because of the flow that is slightly different, or whether there are other aspects that we are still missing in this initial simulation. That is the end of my presentation, and I think we still have a few minutes left for any questions that you might have about either the system itself or the potential modeling aspects. Or
yes, thank you, Viera. If you haven't done so already, you may send your written questions to the group using the questions pane on your control panel. And at this time, I'd like to invite Simulations Plus President John DeBella to the panel as today's moderator. John, please go ahead with the first question.
Okay, well, yeah, thank you very much, Arlene. Let me go ahead, get my camera turned on. Thank you so much, Jay and Vera. What a real treat for the audience today. Two titans in your respective industries describing how validated market-leading technologies can be combined to really unlock valuable safety and efficacy information prior to first-in-human studies. We'll now go ahead and open things up for questions. As Arlene mentioned, you may either type your question in using the questions pane on the GoToWebinar panel, or you may ask your question directly using the hand raising icon in the GoToWebinar system. The questions are coming in fast and furiously. And Jay, I'm gonna tee one up for you to start. A question: what is the benefit of using a microfluidic device compared to conventional in vitro systems?
Well, what you're trying to predict is what goes on in the human body, which is all interconnected. And so current in vitro systems are really not interconnected. What you're doing is you're measuring biomarkers separately and then try to put it into a model to extrapolate function. So one, we have the interconnection and the crosstalk between the different organs, but also the functional readouts are really key here, because most of the current in vitro systems are looking at death as what their marker is, or whether it's leading to death. Whereas we try to avoid death entirely, because if you're starting to get death in a clinical trial, that's really a bad thing. Okay, so those are the two main things why our systems would be better, or microphysiological systems entirely, because we're not the only company out there doing this for these systems. And the other thing too is, I mentioned rare diseases. Currently, there's not, for almost over 7,000 different rare diseases, there is no in vitro models that you can actually use that the FDA would affect, where we can actually build those for the rare diseases. So those are the two main reasons, I would say.
Yeah, thanks, Jay. And actually, kind of building on that last statement a little bit, there was another question. Could you talk briefly about how you would build up and or define some of these diseased or special population environments? There were some questions about whether or not this could be applied, for example, first in pediatric or first in disease state simulations. So could you talk a little bit about what sort of magic would go on behind the scenes to create those systems?
Right, this is where the combination with the PK/PD modeling comes in. We collaborate not only with Simulations Plus, but also with Stefan Schmidt at the University of Florida, you know, where he is actually really focusing on pediatrics in terms of modeling and simulation. And what we can do is we can actually take pediatric mimics, because we can actually do immature tissues as well as mature tissues. and certain things that are very specific to children, or even prenatal, and be able to model those in our systems, to be able to feed into the PK/PD models, to be able to predict what would happen in an adolescent or a children model, pediatric model, that would actually help in terms of informing your IND to the FDA, or actually discussing with the FDA where can I take an existing drug to apply it to pediatrics.
Very good. This may be a question for both of you. It was asked, Jay, during your presentation, and then Vera, you might have touched on it a little bit, but you know, for the examples of terfenadine and fexofenadine both, you know, there were liver and cardiac compartments included in the systems where metabolism due to enzymes in the liver maybe negate the cardiac effect. With these products being given orally, how do you account for the intestinal metabolism and or absorption components? And is this where we bring in the PBPK modeling to define and inform that component to the overall prediction?
Vera, do you want to tackle that first in terms of what your current modeling capability does with GastroPlus, and I can add in terms of what we can do in terms of a modeling version?
Yeah, so in terms of the modeling, we have actually incorporated it in the model itself, right? Based on the in vitro system, we incorporated only liver. We fitted the metabolism in the liver, but then taking the capabilities and the descriptions that we have in GastroPlus, which already incorporates the physiological expression levels of that enzyme between the gut and liver. It allowed us to very easily to extrapolate, right, how the what would be the corresponding metabolism in the intestine. And of course, for the absorption and so on, we used the full ACAD model for the absorption of the compound.
Yeah, and this is the beauty of us working together, in that, you know, we can either put in the GI tract into our heart-liver model, which we've done, or if it's cost effective to do it a different way or something like that, we can use the GastroPlus terminology, because we can now integrate our in vitro PK/PD model with the in vivo predictive models that you guys have been developing for years. Or we can do both, which is add in a GI tract model to be able to look at transport, as well as the metabolitic conversion, because GI tract also has, you know, 3A4, 2D6, other these enzymes in it, and then use that to inform the GastroPlus model to be able to make it even more accurate than what it can currently do based upon the literature parameters.
And let's build on those responses from the two of you. How can we expand this technology and the combination of the HoaC and PBPK approaches to include DDIs?
Yeah.
Well, I would expect that it would be easy to incorporate both compounds in the dosing in the system, right? To give us an idea about how the elimination or the aspects of the compound distributing throughout the system would be affected by another compound included in the system, right? From the modeling point, this would not be a problem, and you know, I'm sure that Jay will correct me, but my guess is that it would not be a problem to include two compounds in that model, right?
Yeah, one of the main things that we're focusing on right now is the whole idea of drug-drug interactions affecting things. We've actually collaborated with another company that's part of this this overall collaboration with Tabula Rasa Healthcare, which has in vivo models based upon patient data, where we're looking at the effect of a compound that inhibits, say, 3A4, and what that does to the PK/PD for, say, another compound, a cardiotoxic drug, an opioid, or something else like that. And we're looking at those drug-drug interactions because it is really, really an overlooked area of pharmacy at this point, which is understanding the effects of different drugs in terms of clearance. And this is a focus area that Hesperos has right at the moment, and collaborating with you know, the in vivo PKP modeling for that.
Very good. There are still a number of questions that are coming in. I think we are going to try and handle a lot of those offline, and Arlene will provide some instructions. But I do want to close things out by asking kind of both of you, what are your thoughts on next steps or future directions tying these technologies together?
Really, it's just a matter of working more closely together. You know, I think that the Vmax question was simply just, it was the absorption of terfenadine is very high, okay, into the systems, and it's about a factor of 100, which accounts for your, I believe, now I got to get my modeling and simulations expert to be able to look at that, but I believe that that's the case. And so just working together with a number of case histories, and again, working with people out in the industry who want to partner with us to be able to solve the problems. You know, if you've got a issue with a compound you're trying to do, I mean, one of the things that's very easy for us to do is work with medicinal chemists. You know, at the lead compound stage where you're trying to do small modifications, but then be able to then scale that up, you have to guess, okay, what's going to be the lead compound, because you're going from micrograms to kilograms, okay, to be able to go into animals or go into clinical trial. Whereas we in these systems, we can measure, say, all 5 of your different candidates for your lead compound, because we only need micrograms in the systems. And then we can then use the PK/PD modeling to be able to predict the clinical parameters or outcomes for all of those, and be able to make a much better informed decision. Because again, you know, depending upon the report, you know, only 11% of compounds that go into clinical trials actually get approved. If we could just double the success rate by getting you better informed at the preclinical level, even the pre-animal level, you know, that will be a huge win for pharmacy. So it's really a matter of just refining the systems by working with people and actually doing the experiments.
I don't think there is much to add to that great description, right? It's just continued collaborations, and this is actually, you know, where if anybody is interested in working with us, joint, you know, collaborations, we'd be very happy to talk to you guys. So.
All right. Well, you know, thank you both. We'll close out the live Q&A session. I again appreciate your time putting the materials together and sharing your knowledge and expertise. Also, we very much thank our attendees for carving out time from busy schedules to learn a little bit more about how we continue to advance modeling and simulation sciences through the integration of novel data capturing techniques. Arlene, take us home.
Thank you. Thank you, Mr. DeBella, Dr. Lukachova, and Dr. Hickman. We appreciate you sharing with us your strategies for deploying this next generation technology. And again, we invite our audience to answer one quick poll and learn more about the utility of GastroPlus for PBPK modeling by visiting our website at www.simulations-plus.com. The webinar has been recorded for playback, and it will be available on our website and YouTube channel. Have a wonderful day, and thank you for joining us.